What if I told you that the very nutrient you have been conditioned to fear, the one you have been actively avoiding for decades, is actually the most critical building block your brain needs to survive and thrive? I know that sounds insane coming from a doctor, but here is what nobody in the mainstream wellness world wants to admit to you. The war on this nutrient has been one of the greatest misunderstandings in the history of modern nutrition, and it may be silently contributing to the epidemic of brain fog, memory loss, and cognitive decline we are witnessing today. Stick with me because by the end of this video, you will understand exactly why your brain is starving for something you have been told to eliminate, and how correcting this misunderstanding could be the single most important decision you make for your long-term cognitive health. Let me tell you about Margaret, 67 years old, from Scottsdale, Arizona. Margaret came to me 2 years ago feeling terrified. She had been forgetting where she placed her keys, struggling to recall the names of people she had known for years, and experiencing what she described as a thick fog that seemed to settle over her thoughts every afternoon. She told me with tears in her eyes that her own mother had suffered from Alzheimer's, and she saw herself heading down the exact same path. Margaret had done everything right, or so she thought. She followed a strict, low-fat diet. She avoided red meat. She had not eaten an egg yolk in over a decade. Her cholesterol numbers were pristine, and her doctor had congratulated her every year on how low they were. But here is the truth that most people never hear. The human brain is composed of nearly 60% fat, and a substantial portion of that fat is cholesterol. In fact, although your brain accounts for only about 2% of your total body weight, it contains approximately 25% of all the cholesterol in your entire body. Let that sink in for a moment. 1/4 of your body's total cholesterol resides in that 3-pound organ between your ears. Does it really make sense that we should be driving those numbers as close to zero as possible? The nutrient I'm talking about today is cholesterol, and the misunderstanding surrounding it has been catastrophic for brain health. Here is where the science gets truly fascinating. Cholesterol is not just some passive structural component sitting idly in your brain. It is an actively essential molecule that your neurons cannot function without. Every single nerve cell in your brain relies on cholesterol to maintain its membrane, that delicate outer layer that controls what enters and exits the cell. Without adequate cholesterol, these membranes become fragile and inefficient. But the role of cholesterol goes far deeper than just structural support. Cholesterol is absolutely critical for something called synaptic transmission, which is the process by which your brain cells communicate with one another. Think of your brain as a massive, intricate electrical grid with billions of connections. Cholesterol acts as the insulation around these connections, ensuring that signals travel quickly, clearly, and without interference. When cholesterol levels drop too low, those signals become fuzzy, slow, and sometimes fail to transmit at all. That afternoon brain fog Margaret described, that struggle to find the right word, that feeling of your thoughts moving through molasses, that is what happens when your neural communication lines start breaking down. The synapse, which is the tiny gap between two neurons where information jumps from one cell to another, is particularly dependent on cholesterol. The membrane of the sending neuron must be rich in cholesterol to properly package and release neurotransmitters, the chemical messengers that carry signals across the synapse. The receiving neuron also requires cholesterol to organize its receptor proteins correctly, ensuring they can catch and interpret those incoming messages. When cholesterol is insufficient, this entire system degrades. You can think of it like a delivery truck showing up at a warehouse where the loading dock workers have gone on strike. The packages arrive, but there is nobody to receive them, sort them, and send them where they need to go. The result is a communication breakdown that manifests in your daily life as forgetfulness, difficulty concentrating, and slower processing speed. Now, I want to be very clear about something. The cholesterol in your brain is not coming from the cholesterol you eat. Your brain makes its own cholesterol, producing it locally through a tightly regulated process. The blood-brain barrier, which is the highly selective gatekeeper that protects your brain from potentially harmful substances in your bloodstream, generally prevents cholesterol from crossing over directly from your diet. So why then does your dietary fat intake matter so much for brain cholesterol? This is where the misunderstanding deepens. While the cholesterol molecules themselves may not cross the blood-brain barrier in significant amounts, the overall metabolic environment created by a very low-fat, low-cholesterol diet can profoundly impact your brain's ability to produce and manage its own cholesterol supply. Furthermore, the transport of cholesterol within the brain relies on specialized carrier proteins called apolipoproteins, and the most important of these for brain function is apolipoprotein E, or ApoE. You may have heard of ApoE before, specifically the ApoE4 variant, which is the strongest known genetic risk factor for late onset Alzheimer's disease. Approximately 25% of the population carries at least one copy of the ApoE4 gene, and these individuals have a significantly higher risk of developing Alzheimer's compared to those with the more common ApoE2 or ApoE3 variants. What few people understand is that ApoE is fundamentally a cholesterol transport protein. Its primary job in the brain is to shuttle cholesterol and other lipids between cells, ensuring that neurons have the raw materials they need for maintenance and repair. The ApoE4 variant is less efficient at this transport function compared to ApoE2 and ApoE3. Think of it like having a fleet of delivery trucks where some are fast and reliable while others are slow and prone to breaking down. If you are someone with the ApoE4 variant, your brain already has a compromised cholesterol delivery system. Now imagine what happens if, on top of that genetic predisposition, you are also severely restricting your dietary fat and cholesterol intake. You are essentially starving an already struggling system of the raw materials it desperately needs. A study published through the Journal of Lipid Research examined the relationship between serum cholesterol levels and cognitive performance in older adults, and the findings challenge everything we have been told. The researchers discovered that individuals with the highest cholesterol levels actually demonstrated better cognitive function, particularly in domains like processing speed, verbal fluency, and abstract reasoning compared to those with the lowest levels. Another study published through Neurology, the Journal of the American Academy of Neurology, followed over 1,000 older adults for several years and found that those with the highest total cholesterol levels had the lowest risk of developing dementia. I am not citing fringe research here. These are major peer-reviewed studies published in some of the most respected medical journals in the world. And yet the message that filters down to the public remains stubbornly one-dimensional and oversimplified. Cholesterol is bad. Lower is always better. Avoid fat at all costs. Let me tell you about another patient, Robert, 72 years old, from Portland, Oregon. Robert was a retired engineer, a brilliant man who had spent his career solving complex problems. When he came to see me, he was deeply frustrated because he could no longer trust his own thinking. He would walk into a room and forget why he was there. He struggled to follow the plot of a movie. Simple calculations that used to be effortless now required paper and pencil. Robert had been on a high-dose statin medication for over 15 years, and his LDL cholesterol had been driven down to levels that his doctor described as excellent. But here is the part of the story that should make every single person watching this sit up and take notice. Statins work by inhibiting an enzyme called HMG-CoA reductase, which is the rate-limiting step in your body's cholesterol production. The problem is that this enzyme does not just exist in your liver, where statins are primarily intended to act. It also exists in your brain. Your brain's ability to produce its own cholesterol relies on the exact same biochemical pathway that statins are designed to shut down. While the pharmaceutical industry and mainstream medicine have long maintained that statins do not cross the blood-brain barrier in meaningful amounts, the reality is more nuanced. Some statins are lipophilic, meaning they are fat-soluble and can indeed cross into the brain. Others are hydrophilic and cross less readily, but the systemic reduction in cholesterol precursors and the overall metabolic shift induced by these drugs can still indirectly affect brain cholesterol metabolism. A review published through the Journal of Clinical Psychiatry examined dozens of studies on statin use and cognitive function, and the results were troubling. A significant subset of patients on statin therapy reported noticeable cognitive impairment, including memory loss, confusion, and difficulty concentrating. These symptoms often resolved when the statin was discontinued, only to return when it was restarted. The Food and Drug Administration itself added a warning to statin labels acknowledging reports of cognitive side effects, including memory loss and confusion. Now, I want to be absolutely clear that I am not telling anyone to stop taking their prescribed medications. That is a decision that must be made in consultation with your personal physician. Statins have undoubtedly saved countless lives by reducing cardiovascular events in high risk individuals, and for many people, the benefits clearly outweigh the risks. But the conversation around cholesterol and brain health has been dangerously narrow, and patients deserve to be fully informed about the potential trade offs they are making. So, what is the right approach? How do we support our brain's cholesterol needs without jeopardizing our cardiovascular health? The answer lies in understanding that not all fats are created equal and that the quality of the fats you consume matters far more than the quantity. Your brain requires specific types of fatty acids that serve as precursors and building blocks for its own cholesterol production and membrane maintenance. Omega-3 fatty acids, particularly docosahexaenoic acid or DHA, are among the most critical. DHA is the most abundant omega-3 fatty acid in the brain, and it is almost exclusively concentrated in the synaptic membranes where neural communication happens. Unlike cholesterol, which your brain can manufacture internally, DHA must come from your diet. Fatty fish like wild salmon, sardines, mackerel, and anchovies are among the richest sources. For those who do not eat fish, algae based supplements can provide a suitable alternative, as fish themselves actually get their DHA from eating algae. Monounsaturated fats, the kind found abundantly in olive oil, avocados, and nuts, are also critical for brain health. Extra virgin olive oil, in particular, contains a compound called oleocanthal that has powerful anti inflammatory properties. Chronic inflammation in the brain is now understood to be a key driver of neurodegenerative diseases, and oleocanthal helps to quench that inflammatory fire. A landmark study called the PREDEMD trial, published through the New England Journal of Medicine, demonstrated that individuals following a Mediterranean diet supplemented with extra virgin olive oil or nuts had significantly better cognitive outcomes over time compared to those on a low fat diet. The Mediterranean diet is not a low fat diet, it is a healthy fat diet, and the distinction is everything. Saturated fat has been the most vilified of all dietary fats, and this is where the conversation becomes particularly nuanced. Your brain requires saturated fat for its structural integrity. The myelin sheath, which is the protective insulation wrapped around your nerve fibers to allow for rapid signal transmission, is composed largely of saturated fat and cholesterol. Without adequate saturated fat, the myelin sheath degrades and neural communication slows down. This is not speculation. This is basic neurobiology. The question is not whether your brain needs saturated fat. It absolutely does. The question is what sources of saturated fat are beneficial versus harmful. The saturated fat found in a grass-fed steak, in pastured eggs, in coconut oil, and in dark chocolate is not metabolically equivalent to the saturated fat found in a highly processed fast food burger made from grain-fed cattle and cooked in inflammatory industrial seed oils. The food matrix matters. The accompanying nutrients matter. The overall dietary pattern matters. Eggs deserve a special mention in this conversation. For decades, eggs were demonized because of their cholesterol content. People were told to throw away the yolks and eat only the whites, discarding the most nutrient dense part of the egg. An egg yolk is arguably one of the most perfect foods for brain health. It contains cholesterol, yes, but it also contains choline, a nutrient that is absolutely essential for the production of acetylcholine, a neurotransmitter critical for memory and learning. It contains lutein and zeaxanthin, antioxidants that protect delicate neural tissues. It contains vitamins B12 and D, both of which are essential for cognitive function. Large-scale epidemiological studies, including research published through the American Journal of Clinical Nutrition, have consistently failed to find a significant association between moderate egg consumption and cardiovascular disease in healthy individuals. In fact, some studies have suggested that regular egg consumption may be associated with better cognitive performance, particularly in older adults. Let me tell you about Frank, 68 years old, from Tampa, Florida. Frank's story is one that has stayed with me for years. He was a retired school teacher, and his wife had noticed he was becoming increasingly withdrawn and forgetful. He would lose his train of thought mid-sentence. He stopped reading books because he could not remember what happened in the previous chapter. His primary care physician attributed it to normal aging and suggested he consider medication, but Frank's daughter, who happened to be a nutritionist, convinced him to try a different approach before going down the pharmaceutical route. She helped him completely overhaul his diet, moving away from the low-fat, high-carbohydrate standard American diet toward a nutrient-dense pattern rich in healthy fats. They introduced wild salmon 3 times a week. They started cooking with avocado oil and extra virgin olive oil. They brought back eggs for breakfast. They added walnuts, flax seeds, and chia seeds for plant-based omega-3s. They eliminated processed vegetable oils and significantly reduced refined sugar. Within 3 months, Frank's wife noticed he was more engaged in conversation. Within 6 months, he picked up a novel and finished it. Within a year, Frank told me he felt like he had gotten a decade of his life back. He told me with tears in his eyes that he felt like he had gotten a decade back. Now, I am not suggesting that dietary changes alone can reverse Alzheimer's disease or other established neurodegenerative conditions, but Frank's case illustrates a principle that is too often overlooked in modern medicine. The brain is not a fixed, static organ that inevitably declines with age. It is dynamic, adaptable, and remarkably responsive to the nutritional environment you provide it. Your brain is still listening. It just needs the right signal delivered the right way. We have to talk about the role of sugar and refined carbohydrates in this equation because the story of brain cholesterol cannot be fully understood without addressing the metabolic context in which cholesterol operates. When you consume a diet high in refined sugars and processed carbohydrates, your blood sugar spikes repeatedly throughout the day. Your body responds by releasing insulin to shuttle that glucose into your cells. Over time, if this pattern continues, your cells become less responsive to insulin, a condition known as insulin resistance. The connection between insulin resistance and brain health is now one of the most active areas of neuroscience research, and the findings are alarming. Some researchers have begun referring to Alzheimer's disease as type 3 diabetes because of the profound role that insulin resistance in the brain appears to play in its development. Insulin is not just a metabolic hormone. In the brain, insulin is critical for neuronal survival, synaptic plasticity, and memory formation. When brain cells become insulin resistant, they cannot properly utilize glucose, their primary fuel source. They essentially begin to starve in the midst of plenty. Surrounded by glucose, they cannot efficiently take up and metabolize. This metabolic crisis triggers a cascade of dysfunction that includes oxidative stress, inflammation, and the accumulation of amyloid plaques and tau tangles, the hallmark pathologies of Alzheimer's disease. Here is where it gets even more interesting and directly connects back to our discussion of cholesterol. Insulin resistance also disrupts the brain's cholesterol metabolism. Studies have shown that in insulin-resistant states, the expression and activity of key enzymes involved in brain cholesterol synthesis are altered. The delicate balance between cholesterol production, transport, and clearance is thrown into disarray. High blood sugar also leads to a process called glycation, where sugar molecules bind to proteins and lipids, including the cholesterol-carrying lipoproteins, rendering them dysfunctional. The combination of impaired cholesterol synthesis and increased glycation damage is a double assault on your brain's structural and functional integrity. Your brain essentially finds itself in a situation where it cannot properly produce or transport the cholesterol it needs, while simultaneously, the cholesterol it does have is being damaged and rendered useless by high blood sugar. The solution to this problem is not to double down on low-fat eating. The solution is to address the metabolic dysfunction at its root by dramatically reducing your intake of refined sugars and processed carbohydrates while ensuring an adequate supply of the healthy fats your brain desperately needs. This is the approach that has shown the most promise in both preventing cognitive decline and supporting brain function in those already experiencing symptoms. The MIND diet, which stands for Mediterranean-Intervention for Neurodegenerative Delay, was specifically designed by researchers at Rush University Medical Center to combine the most neuroprotective elements of the Mediterranean and DASH diets. It emphasizes leafy green vegetables, berries, nuts, whole grains, fish, poultry, beans, and olive oil, while limiting red meat, butter, cheese, pastries, fried foods, and fast food. Multiple large-scale studies have demonstrated that adherence to the MIND diet is associated with a significantly reduced risk of developing Alzheimer's disease, even among those with genetic risk factors like ApoE4. Notice what this diet does not do. It does not restrict healthy fats. It does not limit eggs. It does not promote low fat dairy. It focuses on the quality and source of nutrients rather than on a single oversimplified macronutrient target. Coconut oil deserves its own discussion because it has become one of the most polarizing foods in the nutrition world. Coconut oil is rich in medium chain triglycerides, or MCTs, which are metabolized differently than the long chain triglycerides found in most other dietary fats. MCTs are rapidly absorbed and transported directly to the liver, where they can be converted into ketones. Ketones are an alternative fuel source for the brain, and they are particularly important because they can bypass the glucose transport mechanisms that become impaired in insulin-resistant brains. In Alzheimer's disease, brain cells lose their ability to efficiently utilize glucose, essentially starving even though blood sugar levels may be normal or even high. Ketones provide a metabolic workaround, an alternative fuel that can enter brain cells through a different pathway and keep them functioning when glucose is no longer sufficient. Some small studies have shown that consumption of MCT oil or coconut oil can produce modest but measurable improvements in cognitive function, particularly in individuals with mild cognitive impairment or early-stage Alzheimer's disease. The mechanism is thought to be the rapid provision of ketones to energy-starved neurons. While the evidence is still preliminary and much more research is needed, the biological rationale is sound and the anecdotal reports from patients and caregivers are compelling. Let me address the concern that is undoubtedly on many of your minds right now. If I start eating more fat, even healthy fat, will not my cholesterol numbers go up, and will that not increase my risk of a heart attack? This is the fear that decades of public health messaging have ingrained in us, and it is the central misunderstanding that this entire conversation is built around. The relationship between dietary fat, blood cholesterol, and cardiovascular disease is far more complex than the simple narrative we have been told. Total cholesterol is a very poor predictor of heart disease risk. Many people who have heart attacks have perfectly normal total cholesterol levels. Conversely, many people with high total cholesterol live long, healthy lives with no cardiovascular events. The more meaningful markers, which are increasingly used by forward-thinking physicians, include the ratio of triglycerides to HDL cholesterol, the particle size and number of LDL particles, and markers of inflammation like C-reactive protein. When you replace processed carbohydrates and industrial seed oils with healthy natural fats, you often see improvements in exactly these markers. Triglycerides drop, HDL rises, LDL particle size shifts from small, dense atherogenic particles to larger, more buoyant, less harmful particles, and inflammatory markers decline. A study published through the Annals of Internal Medicine, a meta-analysis that reviewed data from dozens of previous studies involving over 500,000 participants, found no significant association between saturated fat intake and cardiovascular disease risk. The researchers concluded that the evidence did not support the long-standing dietary guidelines recommending severe restriction of saturated fat. Around the same time, another major study published through the British Medical Journal found that individuals who replaced saturated fat with vegetable oils, rich in omega-6 polyunsaturated fats, did indeed lower their cholesterol, but surprisingly, those with the greatest cholesterol reduction actually had a higher risk of death. The cholesterol went down, but the outcomes got worse. How is that possible? It is possible because cholesterol is not the enemy. It is a vital, essential molecule that your body and especially your brain requires to function. When you drive it down by artificial means, you may be solving a number on a lab report while creating a deeper, more insidious problem that takes years or decades to manifest as cognitive decline. This is not to say that everyone should start eating butter by the stick and bacon at every meal. The quality and context of your overall dietary pattern is what matters. A diet rich in vegetables, fruits, nuts, seeds, fatty fish, olive oil, and other whole foods, with moderate amounts of eggs, grass-fed meat, and dairy, and very low in refined sugar, processed foods, and industrial seed oils, is the pattern that has been consistently associated with both cardiovascular and cognitive health in the highest quality epidemiological research. This is not an extreme diet. It is simply what humans ate for most of our evolutionary history, before the rise of industrial food processing and the misguided low-fat dogma that followed. Let me return to the science of myelin for a moment because understanding myelin is key to understanding why your brain needs cholesterol and saturated fat. Myelin is the white fatty substance that wraps around the axons of your neurons, much like the rubber insulation wrapped around an electrical wire. This insulation serves two critical purposes. First, it prevents the electrical signal traveling down the axon from leaking out and dissipating before it reaches its destination. Second, and even more remarkably, it dramatically increases the speed at which that signal can travel. In unmyelinated neurons, signals travel at about 1 to 2 meters per second. In heavily myelinated neurons, signals can travel at up to 100 meters per second. This hundredfold increase in transmission speed is what allows you to think quickly, react instantly, and process complex information in real time. The composition of myelin is striking. It is approximately 70 to 80% lipid, meaning fat, and a substantial portion of that lipid is cholesterol and saturated fat. The brain prioritizes cholesterol for myelin synthesis above almost everything else. Oligodendrocytes, the cells responsible for producing myelin in the brain and central nervous system, are among the most metabolically active cells in the body. They synthesize enormous amounts of cholesterol and other lipids to build and maintain the myelin sheaths around the estimated 100 billion neurons in the human brain. The average oligodendrocyte can support myelin sheaths on up to 50 different axons simultaneously. This is an incredible metabolic demand, and it requires a constant, reliable supply of the raw materials needed for lipid synthesis. When myelin degrades, the consequences are devastating. Multiple sclerosis, one of the most well-known demyelinating diseases, occurs when the immune system attacks and destroys myelin. The results include muscle weakness, coordination problems, vision loss, and cognitive impairment, but even subtle subclinical myelin degradation can impact your cognitive function. As we age, the efficiency of myelin repair and maintenance naturally declines. Oligodendrocyte precursor cells, which are the stem cells that can differentiate into new myelin-producing oligodendrocytes, become less responsive to repair signals. This age-related decline in myelin integrity is now thought to be a significant contributor to the cognitive slowing that accompanies normal aging, and possibly a factor that accelerates the progression toward more serious neurodegenerative conditions. Ensuring an adequate supply of dietary fats, particularly cholesterol precursors, saturated fat, and omega-3 fatty acids, provides the raw materials that the myelin repair system needs to continue functioning as effectively as possible throughout your lifespan. Here is another critical piece of the puzzle that is almost never discussed in mainstream health media. Your brain has its own cholesterol cleanup and recycling system, and this system relies on a protein called CYP46A1. This enzyme converts excess cholesterol in the brain into a metabolite called 24S-hydroxycholesterol, which can cross the blood-brain barrier and exit into the bloodstream for eventual elimination by the liver. This is the brain's primary mechanism for maintaining cholesterol homeostasis, the delicate balance between production and clearance. When this system functions properly, cholesterol does not accumulate to toxic levels. But here is the fascinating part. The activity of CYP46A1 is influenced by the overall metabolic environment of the brain, including oxidative stress levels, inflammatory status, and the availability of certain cofactors. A diet rich in antioxidants from colorful fruits and vegetables combined with healthy fats and low in inflammatory processed foods supports the optimal functioning of this cholesterol clearance pathway. Conversely, a diet high in sugar and pro-inflammatory industrial oils can impair CYP46A1 activity, leading to cholesterol accumulation and neurotoxicity. This is another example of why the simple cholesterol is bad narrative falls apart under scrutiny. It is not about how much cholesterol you have. It is about how well your body, and specifically your brain, is managing its cholesterol economy. We have to talk about phosphatidylcholine because this is another brain essential nutrient that intersects directly with cholesterol metabolism. Phosphatidylcholine is a phospholipid that is a major component of cell membranes, and it is particularly abundant in the brain. It is also the primary dietary source of choline, the precursor to acetylcholine. Acetylcholine is a neurotransmitter that is absolutely critical for memory, learning, and attention.